For the past three lectures or the three chapters which we covered, you know about the details when you have written an exam, and that part is around the okay innate immunity, specific immunity, and then for the specific immunity we did study um, the immunological defense molecule, that is antibodies. So this we covered up in the last class on the chapter 3. But now I'd like to just a little bit of focus. So we'll go through how these antibodies be recognized. So now our antigen is recognized. This antibody recognition of antibody and recognition of antigen. So how the antigen is recognized, and then how this recognition leads to synthesis of antibody. I put it in this way. See, pathogen or any bacteria, that leads to production of antigen. Ag is antigen. Then the antigen should be recognition. And this recognition will eventually lead to antibody production. So this sequence, we are going to study about this, the recognition part. Do you follow that, Paul? So when we want to stop any pathogen spread in our system, we need to identify the pathogen, and also we also studied about the antibodies production. So first we should recognize, as per any enemy territory, we have to identify the enemy. So for that we need to have certain type of patterns and how it looks like, and then what he is, what language he speaks, or the terrorist or the enemy, and how we can tackle those to prepare ourselves to make a defense mechanism. In this strategy, the antigen is our enemy, and then we should know what is the structure of antigen and how this antigen is going to be recognized by our cells. Now we will go on to that step, recognition, and the cells are used for recognition, are the cells that will identify. The cells can see, okay, and then they interact, and then the cells with eventually perpetuate are towards the synthesis of antibody, or how the recognition leads to relay the information. Hey, there is an enemy is being sighted. Relay the information about the sight of an enemy. In this case, it is antigen. So all this part we are going to cover today. Okay, so that's why the topic of today's lecture is membrane receptors. In other words, the membrane is the one which present outside of these cells. Okay, now I'm talking about the cells, what type of cells they are. As I mentioned before, and there are cells, two types of cells. We know about the B cells, that is derived from, historically speaking, is the bursa cells, that is from the birds, and they call it as a B cells, and other type of cells are T cells, thymus derived cells, T cells. Okay, and these B cells and the T cells, they have onto the membrane, I just put the membrane here, this is a nucleus and this membrane. So this membrane will have different types of proteins as the receptors, like here, and then so just I'm drawing this. How this is being organized? What is the membrane is constituted? This is the nucleus, cytoplasm, and this is the membrane. Okay. Our point is how to study these membrane receptors. The receptors, nothing but they are protein molecules, and these protein molecules are organized in the membrane like this. These are the protein molecules. So now I want to know, do you know about what is membrane? It's a biomembrane. Yes, do you know about this membrane? How the cell membrane is being made up of? For your information, just I want to give a review about the biomembranes. And the membrane is made up of, I put another one, membrane, biomembrane, okay, biomembrane, sorry, okay, made up of lipids, okay, and proteins. What is the lipids all about here? You know about the fatty acid. Lipid is made up of glycerol and fatty acids. Okay, the lipids. Glycerol structure, if you know about CH2OH, CHOH, CH2OH. Now here, yeah, CHOH, CH2, 1, yeah. This is for the 4, yeah. Here hydrogen, hydrogen, yeah. Hydrogen, hydrogen. CH2OH, this is CHOH and CH2OH. So this is going to be esterified by fatty acid 1, fatty acid 2, fatty acid 3. So the result is going to be triglycerides. We got the 3 fatty acid esterified with the 1 molecule of glycerol. So this is 1 triglyceride molecule. 1 triglyceride. And this triglyceride, in somewhere, it may happen to have fatty acid 1 and then fatty acid 2. Sometimes this hydroxyl molecule may be phosphorylated, so you have got the phospholipids. This is a triglyceride, and this is a diglyceride with a phospho molecule. So phosphodiglyceride. So otherwise it's called the phospholipids. So this part is a long chain fatty acid, which is a hydrophobic because of the hydrogen and with hydroxy group, which call it as a hydrophilic nature. So in short, I can write like this. This is one molecule, this is another molecule. So this is hydrophilic and this is hydrophobic. So if I draw like this, that is one layer of the membrane. Like this, I'll draw in a different way. Like so, this is one layer, okay. And then the another layer at the bottom here, and it will go like this. So what do you think on this particular structure? Is it made up of lipid? This is one layer and two layer, so it is called as lipid bilayer membrane. Do you follow that? So this is the strategy of all this membrane is made up of on the biological cell. It goes like that. So you have a lipid. is surrounding this membrane, and here the nucleus is there. So what happened here, the protein molecules are going to be intervene here. Suppose here it comes out like this, the protein molecule. It occupies like this part. And then here some protein molecules. And here some protein molecules. And here this protein molecules. Here this protein molecules. Here this protein molecules. And sometimes you also the cholesterol which is present inside. So the lipid bilayer membrane is made up of proteins and lipid bilayer here, the lipid moiety, otherwise you got the fat, otherwise you call it as a lipid, phospholipid in general. And these proteins, the function of the protein is a transporter protein, or this protein act as a receptors, act as receptor. Receptor of what? There are several receptors here, but here the receptor, in this case here we talk about the antigen. You follow that? So membrane receptors, that is our today's topic. The purpose of the receptors is to receive or recognize the antigen. The antigen is coming from the bacteria or virus or any other pollen or fungal or any other pathogen getting into your system. So when you see the cell, and that cell is a B cell in this case. So B cell, and this is a receptor. In other words, we call it as, in short, BCR, B cell receptor. The same thing which we can also study about the T cell. So the T cell receptor is going to be TCR. Okay. So we are concerning about this molecule, this protein of this receptor which is present in the lipid bilayer membrane. And this protein, which we will study, if you see the structure, is that it's present at the surface level. This is the surface outside, okay, and in between that we call it as a transmembrane. I'll write it in a different way. So this is the lipid bilayer membrane, and this is the protein. So this protein, which will connect the outside of the cell and inside of the cell. Inside you have the cytosol, and this is the nucleus. Okay. So this receptor molecule is connecting outside and also as well as the inside, and in between that you get a membrane domain. You call it as a transmembrane. So in the protein, we normally say this region as a transmembrane domain is nothing but the region of the protein which is present over there. So you have one part here inside and another part is outside as a surface and the transmembrane domain. So now I am talking about the structure of the receptors. The receptors are present in the membrane, and the outside is a surface receptors, and in between the membrane, and then inside in the cytosol. So whenever, in a nutshell, in a summary, whenever there is an antigen which is going over there and it can recognize by this, we will see how it recognizes here. And as soon as it recognizes, it passes it on the signals over into the cytosol, and then where it can be transduced with the other molecules, and thereby the message is passing on to the nucleus, and the nucleus is going to activate certain genes, and that gene will eventually produce antibodies. So we will see this pathway, how and different stages of this one. So that's a part of this membrane receptors. Okay, do you follow that principles? Yes.
That is the same thing of the transmembrane domain, that's the integral protein, right?
Yes, that's the integral protein. It is not a separate, the same protein which is there, but the synthesis, we are going to see the details of this, because our concern is about this molecule today. The entire class we are going to discuss about it. That's why I'm going in deeper. So you to recognize where the molecule is there on the membrane, and how the molecule it looks like, and which part it's going outside and inside. I mean, our major task is to recognition. Once it recognizes, it should pass it on to the nucleus, that information. So nucleus will not know. It's a blinded. It can act when it get the signal. That's all. It cannot sense anything, but it can act according to the signal which is being coming from the surface region to the nucleus. So we will see that part. Okay. So this system, we call it as a cunning system. I put it in that way. That's what in the textbook also there. Cunning system. What is cunning system here? Whenever the antigen is coming here, and this is basically is a virgin cell, it will not know anything about it. But there is some complementarity. Something about this antigen can bind to these receptors. This is not an antibody. Just please remember that. This is not an antibody, but it's only the membrane receptors. And these receptors can bind with antigen. Imagine it is nothing to do with any of the antibody, but there are certain region of the protein which can recognize a likely antigen-like a protein which is foreign to that one. It is not recognized by our body, so it can be recognized. So that's the situation here. This antigen can be recognized by the receptors. So you may ask how? There's no antibody, but in our inherent of our defense mechanism, that's the beauty of it. Already we have certain type of antibody-like protein. It is not antibody, but antibody-like proteins that called the receptors, which is already there in different B cells. It's already there. Any type of antigen, it can recognize a little bit. Not whole of antibody, but little bit it can recognize on this part. One or two, not all the cells will recognize. Only few cells can recognize. That's why in the virgin B cells, if you have more of B cells, it can recognize. That's antibody. And then as soon as it capture one or two antibodies, these cells, hey, come on, yeah, I'm waiting for you, I receive you, just like that. And then eventually these cells, this antigen will bind, and then it will photocopy that one. Okay, this antigen, as soon as it's bind to it, and our bacteria, and bacterial membrane or the surface receptors, they can bind with this. As soon as it binds with that, it passes it on the signal, and this signal will produce the antibody, and finally what will happen, this lymphocyte attract, it pass it on the signal outside, and then phagocytosis, and this is going to be the antigen or bacteria is be eventually killed. So the cunning system means it cheats the bacteria or antigen, and thereby it leads to its death. So that's a thing. It's a very cunning system in that way. So antigen can be led to its doomsday, you can call it. Okay. So it select. So there are several cells. I'll put it in that way. There are. I'll put these are the cells. Okay. These are the B cells, and these B cells. Some of the B cells can identify this guy, this one. Then as soon as it identify that, and it can also synthesize an antibody. It recognizes, but we are going to see that the steps involved. Okay. How this will binding with this one. As you know very well, the complementarity, right? Complements. Complement shape of receptors. Okay, I'll put it there, this one as a cell, and this cell will have like this, like this, like this. Okay. So this is a receptor. It doesn't know about it, the structure of this antibody or anything, but it randomly organized. In another B cell, you get the receptor will like this, it will go like this. In another B cell, the receptors will like this. Another B cell, the receptors goes like that. So you have a different population of the B cell and the B cell, the membrane, which have the different type of receptors hanging around the cell. So now there is an infection, and that infection of the bacteria will produce an antigen. The antigen shape is something like this. This is antigen. And this antigen shape is something like this. And the receptor is like this. If this B cell will get in touch with them, then immediately it can bind to it because of the complementarity, or it attract this, and this is being attracted by this. So there is an interaction here and here. It cannot bind here, and it cannot bind this, and it cannot bind over this. But this one suits the complement shape, and thereby it can bind. Without any antibody, without any help, we don't know anything about it. That's why I said put virgin B cells. The virgin B cells mean it never exposed to any other antigen. But it is in our system from the germline, from the birth onwards. The B cells, the different population of B cells, and the membrane, they have the receptors. That should understand. That's what I'm saying. So once these interactions here, it combine, and then it can fit into it, then the signals will go on. When the signals will go onto the nucleus, and this nucleus will start proliferating. So this result is proliferation. It activates the gene. Then you say, how it will proliferate? These receptors is already, this protein is already has a gene. Do you understand that? Yes? This receptor is a protein, and this protein has a gene, correct? When the synthesis of the cell, this gene is already activated. That's why you get this protein which is sitting onto the membrane. Is that right? Otherwise, you won't get this protein into the membrane. Already the gene is expressed, correct? So when you get the signal, eventually that signal will go and activate this gene. It activates the more of the gene. At the same time, it also activates the cell division. Since this particular B cell predominant of these particular receptors, and the activation of this gene as well as the cell division will lead to a clone, one particular type of cellular expression of that gene, one type of antibody. There's a one type of cells will be, because this cell started dividing it. Initially it was a virgin cell, and now as soon as it contact with that, it contact the signal, and the signal says to the nucleus, hey, there is similar protein like me, like I can combine with another guy. I need help. I need more of these particular antibodies, or not I mean it's an antibody, but I'm saying more amount of the protein I need it to tackle this guy. I identify this guy. So that is the relay of this information. When this relayed to this nucleus, and the nucleus activate the gene, and that gene is eventually synthesized of more, but one cell cannot produce all of them. So what will happen? It activate the cell division, and the cell division will eventually get into more of these particular cells. When it mature, and these cells we call it as plasma cell. Plasma cells is nothing but a B cell, which a matured B cell, which will synthesize one type of antibody. So now you get this antibody is being released into the system. We will see how the structure of these antibodies and then immunoglobulin from this will be secreted out. And thereby you get the exposure of the antigen that lead to cloning, a type of protein, a type of gene, a type of cells, a plasma cell will start emerging out. One particular type of antibody it will produce. Do you understand that? How the membrane receptors of B cell will interact, and thereby it will induces the plasma cell. We will go into that little bit of that mechanism now. Okay. Something like the plasma cells, the one with the B cell, it gives the photocopy of that. Because of the gene already has been expressed, the mRNA and everything is there. So this activation of particular protein, this gene expression is being further activated because of the demand of more of the demand and the signal, it will activate the cell division. If there is no antigen, what will happen? This cell remain as a B cell. It will not activate. It's keeping quiet. It's a dormant cell, and it is sleeping cell. It is not going. It comes and birth, and then as soon as if nothing happens, it will die and go off. That's it. But when and where the type of antigen binding, then that cell is awakening signal. Hey, don't sleep. Now this is your time to divide and produce more of that particular receptors. But it will also produce the receptors at the same time. This receptor molecule will eventually increases the shape and that certain type of protein which will go and bind to the membrane receptors, and certain type of protein, the same antibody will secrete it out. Now we shall say how a same receptor molecule act as a reception, at the same time the same receptor molecule in a different way it can secrete into the circulation. The same gene, just I want to make sure, a same set of genes. That genes will express, I'll put it that way, in a different way, same gene, but this gene will in one form, it can act as a receptor protein, and the same gene, which is the product of the gene, it will go for antibody. And this antibody getting into circulation, and this receptor molecule stays in the membrane. Yes? Do you follow up to this point? So this gene, when this gene is activated, whenever there is an antigen plus receptor, this complex signals relay the information, and then this gene is activated. This gene is what we call it as a receptor gene, receptor plus antibody genes. This gene. So this is being activated, and thereby a part of them is being processed. This process, how same protein, but how they form as an antibody, another one is a receptor. We will see now the next step. Okay. So far, any questions in any center? Cinco Ranch? Or in Victoria? No? No questions from Cinco Ranch? Okay. In the area, they studied about the anti-IG. That's one experiment. I'm just getting in on an experiment for you. Okay, the science is full of an experiment. The results of the experiment we are studying in the textbook, right? Experiment. The anti-immunoglobulin, anti-IG study that revealed the membrane IG, the membrane part, okay, membrane integral membrane part, membrane IG, and that is to be a type of class, IgM class. I want to recollect what we studied before, IgG, IgD, IgE, IgM, like different type of immunoglobulin molecules in our system, in our blood. Yeah, right? We have studied in an earlier class of what type of immunoglobulin we have. So the experiment, in the earlier experiment, that revealed the membrane, which I mentioned before in my another slide. See, if you recollect this picture, this membrane part, this part of the immunoglobulin, that is made up of, our transmembrane domain of that protein is made up of IgM class. This is IgM class. Okay. So the antibody specificity lies in the individual rearranged genes. That is, this is IgM transmembrane, but this part, the antibody recognition part over here, right? are, this is the transmembrane domain of IgM class. And the antibody recognition site are the antigen recognition or antibody specificity that is lies in VJC genes, which I mentioned before. See this gene, right? The gene which is going to be acting as a receptor as well as an antibody. On the receptor side, in mostly you get the IgM class because that stays in the membrane part of that. And then the antibody, which is having both this membrane part as well as the another part, we will see that one. So the VJC, and they have also the another subtype is a kappa and lambda and delta genes. There are different type of genes. So the cell surface Ig, cell surface of the membrane receptor, right? It's formed due to differential splicing. Differential splicing. What is differential splicing? It's a little bit of molecular biology, and just I want to get into the system now. I want to explain this part. So far, if you recognize what we studied, the gene which is responsible for producing a receptor, at the same time it can also produce the antibody. So the mechanism I'm going to explain to you now. Through differential splicing, the mechanisms through differential splicing. What is this? We will see that. In short, the story starts with one cell, that the B cell, T cell, or here in this case B cell, one B cell, that will produce one plasma cell. Do you agree that one? As soon as it receives the B cell receive the antigen, and the antigen receptor interaction, that changes it, mature or awaken this cell. The B cell is sleeping, and as soon as it binds with the antigen, it started awakening. Oh yeah, it's an alarming, just like an alarm when you get up in the early morning. So this is an alarm for them. Otherwise it will sleep, it will go on. So it will awake the cell, and then it will immediately changing into the plasma cell. And this plasma cell in action now, okay, it will produce one type of antibody, one type antibody, okay. So when they produce this antibody, or the immunoglobulin in that case, some of them, okay, some of antibody or antibody protein, I put it as a protein, antibody protein, go to membrane surface. Some of the antibody protein go to the membrane surface, okay, and here it act as, the function for this one, function as membrane receptor, okay. And then this is one part. The another part of the same thing here, the one type of antibody here, and some of the antibody, second type, this is type 1, I mean the not type 1 is the same, one group of protein, it goes and stays there, another group of the antibody protein, some of the antibody protein that go to blood or circulation, go to circulation, okay. So in this case, I'll put it here, one gene, some of the product, the gene product will go and stays in the membrane surface as a membrane receptor. The same gene and the product which is coming out here, and some of these antibodies secrete the antibody that go to the blood and circulation, and then here we call it as a secretion. So secretory protein antibody go to the circulation. These are the stays into the membrane. These stays back onto the membrane itself, so and act as a receptor molecule for this antigen. So both receptor can also bind to antigen, antibody, secreted antibody can also binding the antigen or the bacteria or viral, whatever the protein is going to be. So this part is the recognition part. So both of them derived from same gene. Do you understand that concept now? It stays in the membrane, this part of this antibody, and this part of the antibody is getting into the blood. Yes? Something like the same police force, which is the homeland security, one side, and another type of a defense you are sending outside your country to get enemy outside. That's what the United States now doing. You know, our soldiers are going outside. They also train how to shoot and how to do, and both of them doing the same function. Some of them are the internal and some of them outside. But both of them are working on recognizing the terrorist. One is the inside the terrorist as well as they are outside the terrorist. So the same thing with the antigen, it act like a terrorist, which is coming outside the cells. Yes, it will go, and wherever the circulation, yes, it will go. And the membrane level, so this is the membrane level, which is going to be internal, which stays in the cell and recognize wherever this antigen is coming up. The same thing is in a secretion, wherever the antigen is going through the circulation, and this antibody will go and capture that one. Antibody go and bind to this antigen. So now this antigen specificity in this case. Do you follow that part? So this in between the gene, to some of them going membrane, some of them going to the antibody secretion, and this part of this mechanism we call it as a differential splicing. Differential splicing. Okay. How this differential splicing will work? In your textbook, you can go on the figure 4.1 in the old, 10th edition. I'll go on to that one here. Okay. Also in a PowerPoint, but I just want to go here. Could you see here now? DNA, that's a gene. VDJ, and here a set of micron units, and as well as here you have a mu, M1 and M2. When the DNA is transcribed, do you know about the molecular biology a little bit? Where the DNA converted to the mRNA and the mRNA into the protein, right? So now the DNA is transcribed and the transcription, and here the splicing will occur where mRNA is VDJ, C mu, and the M1. What happened here? This part is being removed during the splicing, but you have one part which is coming out M1 and M2, C1 and 4 up to this. You know, it will come on VDJ, C up to this. It comes out M1 and M2 in one the whole, and then another part is VDJ and C micron 1 to 4. See, up to this one, and the whole part is another one. So that's the way that it will splice them out, two of them. And one is the coming out the membrane. If this mRNA is becoming a protein, that form IgM. But here it also produces these also IgM, but it lacks the M1 to that's the membrane part. If it translates into a protein, it stays in the membrane. If this particular gene is not expressed as the protein, when this protein M1 and M2 is not translated into the mRNA, this mRNA is translated into the protein, you see that M1 and M2 is missing here. So because of this lack of this membrane component, it cannot stay in the membrane, so it is secreted out in the blood. A type of protein which is required to stay in the membrane, that particular protein is lacking when they transcribe. Same gene, the same gene, but some of them is expressed with the membrane component and some of them is lacking the membrane component. So if that one gene, it will give the two product of protein. One gene will produce two protein. One protein stays in the membrane, another protein cannot stay in the membrane, so it quit the cell. It will go out of the cell and go to the circulation. So that's why the one which is going outside the cell because of the lack of the membrane component of the protein, that is called antibody. The one which stays in the membrane at the surface of the membrane that act as a receptor. But you should remember that the gene of the antibody will have one gene, but it have a two different product. The same gene will have a receptor and same gene which will go as an antibody. Why is the same gene? Because the function of this defense mechanism to bind with the antigen, it can recognize the antigen. So you need the same type of protein to recognize, and the membrane receptors, as well as the antibodies going out, it should capture the other antigen which is circulating in the blood. So you have two proteins but one gene, so that it can do the same function of binding the antigen. Do you got the concept? It's a bit difficult, but once you understand the principle, then it is easy to understand. Okay. So the another one, the surface membrane IgM and IgD receptors. The another part is the 4.2. You have the same thing here. Here, okay. In this DNA and primary RNA transcript, you get the VDJ-CC5, and here this is being ruled out. This is being out here, the dark one. I mean, it's coming over here, and then C micron, and then C gamma 3. C gamma 3 is be out. But what happened here? The splicing VDJ-C mu up to this, and then VDJ-C delta, which is coming over here. And this translation into membrane IgM, and this is a membrane IgD. The membrane IgM, you got C micron, and then here the C delta. So VDJ-C delta, if that is being translated into the protein, that become IgD. But when VDJ and C micron, that is getting into IgM. So this is another type of splicing. So my point is how one gene make two protein. The answer is by differential splicing. So if I ask a question to write a short notes on differential splicing, see, the question is, what is differential splicing? If I ask that question, okay, so the answer is, the answer for the differential splicing, the answer is one gene, two protein. If I ask the reverse of two protein, and the function of the two protein is one is the receptor, another one is the antibody, and what is the gene? What is the mechanism involved of these receptor and antibody synthesis? That's the another way which I ask the question, but the answer is the differential splicing. So either way, if you understand the differential splicing, that is the answer that you have to give. And whenever I say differential splicing means one gene will give two protein in this case, right? The function is the same function. One gene, one function, but two proteins because of two different nature where it is. So that's the part you should understand. Okay? So the B cells is being awakened to form plasma cell, and these plasma cells will produce more of antibody, that secreted antibody, and some of this protein which is also stays there, right, that also have receptors of B cells. It stays there. It won't secrete it out. It stays there, the cells itself. Also in the B cells. So in the virgin IgM, because it is a membrane component, virgin B cell. Do you remember that virgin B cell which is having an IgM component? Because that's why it's the membrane component, and it bind with the antigen. The virgin B cell has IgM. So you have a little bit of a mature one, mature or awakened the cells, okay, that cell will have IgM because of the membrane component. Also IgD, the one which I just showed you before. So it gives another splicing IgD which will have. And then if you have the fully mature B cell, you have fully. Let's go here. Okay, fully mature cell. Fully mature cell B cell will have IgG. So when you collect peripheral blood and want to separate them out of the cells, if you put antibody to anti-IgM and anti-IgD and anti-IgG, so in that case you can easily separate them out. If I put the anti-IgM will react here, so the antibody which can isolate this. Then the mature cells, if I put an anti-IgD, these are the antibody specifically. This is the antigen, this is the antibody. So it can bind together and isolate. Then anti-IgG, this is an antibody for this. So if I put in a peripheral blood, so I can separate them out when I put this mixture of this. So it can specifically bind to that particular cell, and then I can separate them, I can study. So that is for our how we can study this particular cell. You may ask, hey, there is a lot of cells hanging around in the blood. How can I study this particular B cells? How to study? This is one of the technique which we will study on our class, maybe on the chapter 6 or chapter 7 on the immunocytochemical or immunochemical techniques. In that we will see about the mechanism of isolation of these cells. So what I'm saying here, the virgin B cells, and then you have mature cells and fully mature cell, they express different type of Ig class, IgM, IgD, IgE. That's what you should know. So if I ask a question, what Ig is being expressed in fully matured B cell? If I ask that question, your answer is IgG. IgG, fully mature cells, okay. If it is which class IgM is expressed, what cells the IgM express? If I ask that question, IgM expressed in virgin B cell as well as mature, half mature. I put half mature B cell, you have IgM there. But where do you find IgD is expressed? Which cell express IgD? If I ask that question, IgD expressed only in mature B cell. Not fully, but mature, half mature cell. Okay. Now I'll go on to the signaling part. Okay. Signaling. Any questions so far? What we have studied before is a B cell, and that B cell will produce the plasma cell, and plasma cell responsible for the antibody. Now, our question is how the B cell, okay, receive signals or processing the signal, and that will lead to, the signals lead to nucleus. So we are going to study now on this part, okay. As I mentioned before, in the cell membrane, this cell membrane, okay, is going on like this, okay, this whole cell membrane, lipid bilayer membrane, and you have the protein, and this protein is a receptor, and this receptor, and in here you have an antigen. So the antigen is binding here with the receptor. So as soon as it make a contact, okay, as soon as the receptor making a receive antigen, or it binds with the antigen, it will activate the signal. So that's why it is as I said, receiving the signal, or otherwise the reception or binding of this antigen. When it binds here, it will convey the message here, the nucleus. But but in the membrane, you have only three amino acids on the cytoplasmic region of the IgM domain. This is the IgM. Remember the membrane part, which is of this receptor, which is called IgM part, right? So it lacks these only three amino acids. It cannot convey the message. We need a larger molecule that will convey the message. Okay. So in the membrane, you have Ig alpha, Ig beta, Ig alpha, Ig beta. I will draw in a larger diagram, easy to understand that part. Okay, now that's a bit of waiting. Okay, great. So here you have the nucleus. There's a binding, and this part is a three amino acids, so it won't. These are the membrane receptors. Instead, you have another type of Ig molecule called Ig alpha, Ig alpha, Ig beta, Ig alpha, Ig beta. Another type of molecule, immunoglobulin molecule, which is adjacent to this receptor. This is the receptor. Okay. It can bind with the antigen. And this antigen cannot bind here, it cannot bind here, it cannot bind here, it cannot bind here. But at the same time, it helps, this protein helps in binding near to the receptor. As soon as it binds here, and there is some changes, structural changes which is occurring into this one, and this protein, this Ig alpha and Ig beta, they have tyrosine kinase enzyme. The kinase, whenever you study kinase enzyme, that is acting as a phosphorylation, phosphate group, okay, for phosphorylation, addition of phosphate. That's what is happening of the phosphate. So this is the high energy phosphate group. The phosphate is another way, it's a language, the language of signaling language. What? Phosphate is like a, whenever there's a phosphate here, it's roaming around here, roaming around in the cytoplasm, it is already there. But as soon as it binds here, the antigen binding with the receptors, this molecule, Ig alpha, Ig beta, this side, Ig alpha, Ig beta, they combine together because they have the kinases enzyme. These kinases will activate this phosphate group and they phosphorylate. So as soon as it phosphorylate, and it will activate other signaling molecules, tyrosine kinase, which is already here. There are other molecules which is hanging around in the cytoplasm. It will activate. Once this phosphorylation, it occurs. Another one. But this group is, we call it as a part of the protein. This part we call it ITAM. ITAM. What is ITAM? We call it in other words, call it like a immuno, okay, receptor, because it is a receptor, tyrosine based activation motifs. Oh yeah. ITAM, immunoreceptor, because this is the antigen and receptor, this immunoreceptor, and the tyrosine kinase, which is here, which is underneath of on the cytosol phase of this alpha, and this activation motif, this particular type of protein. So they are called, this part is called ITAM. So, and they are responsible for the, convey the message to the nucleus. So now we are going to study the function of receptor. The function of the receptor, what this receptor will do, B cell receptors, it receives the signal, but it cannot activate by itself. But that protein is lacking the ITAM. So the tyrosine kinase is not present in the receptor, but it needs the help of the neighboring protein. And the neighboring molecule is Ig alpha, Ig beta, Ig alpha. So neighboring Ig alpha, Ig beta, they will be activated as soon as it gets, because they combine together and the signal is being converted to this item or this protein kinases. That is the main reason it will go on bind and then to the nucleus and convey the message, and the nucleus started transcribing a particular protein. So this is called item. So for why I showed this slide, you should understand the crosstalk of antigen antibody, and that is going to be translated in the form of phosphate molecule, and that molecule we call it here the item, immunoreceptor tyrosine based activation motif. This is another question which I will ask for your exam about it. What is item? Okay. Now, the next type of receptor we have to call it as the T cell receptor. What I'll do, I'll give you a short break of 5 minutes and you can come back and then we can do it further. Okay, and do you have any questions so far? Any question? Sync orange?
So far, so good.
Okay. So only 3 students over there in sync orange? How many? What happened? Any exam tomorrow, today? No? Okay. So the next part, we will look into the details of the T cell receptor. So the previous one is the B cell receptor and how that receptor is activating the other pathways in the signaling mechanism, and as well as the plasma cells and plasma cells into the antibody. And now we are going to look in the details of the T cell receptors, or the T cells. And these T cell surface receptor. Okay. They are also as a transmembrane or heterodimer. They also present as a transmembrane heterodimer. I want to explain this a little bit. What is transmembrane here? The transmembrane is in the cell and this nucleus and this part. This part we call it as a transmembrane, just like IgM in a last case. This is a transmembrane. And the heterodimer, dimer means 2, okay, and the 2 proteins, this type of transmembrane will have 2 proteins, and they have not similar protein. We have 2 dissimilar protein. All right, it's a same type of protein, we call it as a homodimer, same or similar protein. But here it's not the same one, or it's a different type, so we call it as a heterodimer proteins. So whenever I call it as a TCR or T cell receptors, which will have 2 proteins, and the 2 proteins are not similar, so they are different. So it is called a heterodimer. Okay, what type of this heterodimer, and they are membrane bound molecules. This dimer we call it as an alpha and beta chain. Alpha is one protein and beta is another protein, and they linked together by disulfide bridges. Disulfide bridges. Not one, there are 2 disulfide bridges. They combine the suppose this is another protein alpha and beta, and they've been combined with this one, two type of disulfide bridges, this alpha and beta chain. So this is the type of protein which is present in the TCR. Okay, alpha and beta. Now we will see how these T cell receptors and this protein is responsible for the binding to the antigen or activating the function, the antigen recognition signs. Okay, you follow up to this point? So T cell receptors are heterodimer, two protein, and they got alpha and beta. So we'll see that one. Alpha chain consists of two immunoglobulin like domain. It's not like a immunoglobulin. It is not immunoglobulin, but alpha chain, two immunoglobulin like domain. And the same thing is the beta. They consist of two immunoglobulin like domain. So TCR, the T cell receptor, is made up of let's go that way. Okay. Now T cell receptor, it made up of two chain, otherwise we call heterodimer, transmembrane heterodimer. One is alpha, another one is beta. Then this alpha consists of two of immunoglobulin like domain. This it's not immunoglobulin, but immunoglobulin like domain, like protein. So in total, how many you have a TCR molecule or the protein will have a 2 plus 2, 4 Ig like domains. Okay, 4 areas. Okay. So in this one, alpha and beta, they have two type of region. Like this is one, this one. Okay, this is for alpha, and this is for beta. Another two, the domain, and they have this region of this alpha and this region. Okay, and this is more significant. Okay, so among these alpha 2 Ig domain like this, one part of this alpha as well as the beta of this part is a variability region, meaning they are the binding region, binding to antigen. This region of one here, alpha 1 and beta 1 chain. And then the another chain which is coming over here in this part, in this chain. Okay, this is the invariant. This is also invariant. So one part of alpha, one chain of alpha is a variability, one chain is a variability region as well as for the beta, one alpha, one beta. The another chain of this is not variable, they are invariable region. These are invariable region. So one alpha chain and one beta chain is responsible for the binding to this antigen, and they are present in the TCR. Do you follow up to this point? I'll summarize. We studied last class about, just a part of this class before we study B cell, and this is another part which we are going to study, the T cell. Okay, B cell and T cell is the part which we involve in the immunological competent cell or immunological cells. We want to study where it will bind with the first line of defense like antigen. So we need to study about the T cells and B cells. When I was in a graduate school, when they asked about the, then it's a joke like T cell. What is T cell? Because T cell, T cell is a D cell, and B cell is a battery cell, right? So that type of things which we want to recognize. I mean, if you want to study the immunological cells, what type of cells you have to remember that. So B cells and T cells, they have receptors, and the receptors which will receive, the antigen. Is there any Jim? Is there anything you want to see? Is not able to see anything?
It's kind of blurry.
Oh, okay. Okay, I'll explain to you now. This is the T cell receptor, which is a surface receptor. Okay. Let me turn off this. Could you see now, Jim?
A little bit better.
Okay, okay. I turn off the light probably. That's the glare. Okay. And the transmembrane heterodimer. The transmembrane heterodimer is the name or the quality of this protein. Transmembrane is the membrane, transmembrane here. And heterodimer means, dimer means two proteins, and two are dissimilar protein. That's why it is called as a heterodimer. If it is the same type of protein, we call it as a homodimer. So it's a heterodimer. And then each protein, like a alpha and beta chains, which is being linked with two type of disulfide bridges. And this is the constitution, or this is the morphology, or this is the structure of this protein, TCR, T cell receptor. What is the function of T cell receptor again? It will bind with antigen. So when it binds with the antigen, we have to recognize what chain which is responsible for that. So if you split into alpha and beta chain, and each chain, alpha chain consists of two Ig-like domain, and beta chain also two Ig-like domain. I mean two proteins, again Ig-like, it's not Ig but Ig-like domain. Among these, one of these alpha chain is a variable region, where meaning that is the region which will bind to the antigen. And here is one of the beta chain, also the variable region. Whereas the another chain, they are invariant. So totally you have 4 chain in the T cell receptor, right? Do you follow that one? T cell receptor, there are 4 chains. And then alpha chain 2 and beta chain 2. Among the two, one is being recognized, one is the variable region, and the beta chain is one among two, one is responsible for the binding. So if you remember the antibody structure, I just draw the antibody structure. Okay, light chain and a heavy chain, right? You may recognize. Only one particular region of that hypervariable region. Rest of them are the constant region. So we call it as the FC region, and these two region which we call it as a Fab, antibody binding region, Fab region. Do you remember that? Because of the capacity to bind with the antigen, this region, we call it an antibody binding region. Otherwise, this region of the antibody fragment AB. Do you remember that? Immunoglobulin fragment AB. So the TCR, T cell receptor, alpha and beta chain, they have similar, because I mentioned before in the previous slide, this region is a variable region and this is a variable region of this receptor. They act just like antibody binding region of fragment AB. Do you follow that? Because the function, last cell, B cell, we have two types of things. Like one is stays in the membrane, another one is going on to the synthesizing of antibody, secreting the antibody, it's going out. But both will bind to the antigen, correct? The same thing is here, but the TCR, the receptor, it is not, the T cell is not responsible for secreting antibody. That you should remember that first. Only the B cell is responsible for the antibody production. B cell will convert to the plasma cell and not the T cell. Yes? We know the two type of cells recognizing the antibody. One is the B cell and T cell, And B cell is give birth to plasma cell, and plasma cell produce more of antibody. But T cell is just act as a receptors, and its function is a bit different. We will see that one. Okay. And here also the alpha cells and beta cells, alpha and beta chain of the TCR, that looks like the binding region of antibody recognition site. Okay, this fragment closely resembling the IGFAB or the fragment AB, and this region is contact with, I put it in this way, so this is alpha and this is the beta chain, right? And then here this region and this region, I put it. And they are responsible for the MHC, major histocompatibility MHC. We are going to see in a little bit. MHC binding region. So I draw a cell. This is T cell nucleus, and you have the region. I put like that. So these two are beta and this is alpha. This is the receptor, okay? These are the receptor protein of the TCR. This region and this region is being responsible for the binding to major histocompatibility. Major histocompatibility, yes. So we call it MHC. I'll call it as MHC molecule, binding to MHC molecule. We'll see that. This is another type of protein where we will see that one. So this T cell contact with MHC. The region is this region, the variability region, alpha and beta. Fag region. So there are two classes of T cell receptors now. Okay, there are two classes of TCR. One is with the alpha beta TCR, that is called TCR2. Another one is the second one is the gamma and delta TCR1. So if I say TCR1 composed of gamma and delta and alpha and beta, and that's what TCR2. So there are two classes of TCR. So if you isolate in the blood, in the human blood, okay, and you will find gamma and delta cells, meaning TCR1 cells, TCR1 cells, the receptors 1 cell, TCL1 cells, they are present 0.5 to 15% of T cells in human peripheral cells. And these cells, the TCR1, these T cells are present in intestine, epithelial cells. They are present in the epithelial cells, that population, as well as they are also present in skin. To what is the function of the T cell? It will bind with any antigen. If there is anything which is a bug which is getting into your intestine, the TCL or T cell receptor R1 or gamma and delta, and they bind with those antigen, which is present in the skin as well as the intestinal epithelial cell. And in the blood, you get 0.5 to 15%. They are more abundant. These cells are intestinal epithelial cells as well as the skin cells is present there. But in the ruminant, the ruminants, the animal kingdom, where the rat and rabbits and those, you get 30 to 80% the blood cells. They are composed of this TCR1 population. Okay. Okay. The next part is the TCR resembles or similar to immunoglobulin IG. Now we will see the morphology. Morphology, how it looks like, this TCR. These molecules similar to immunoglobulin. They are not immunoglobulin, but they are similar to immunoglobulin or similar to IgM, IgD, IgG, like that. It's similar to that one. And the T cell receptors, the beta chains, they follow the similar arrangements. See, the arrangements is coming similar to, I put similar to, meaning it also follows the same rule like the VDJ region and also a constant segment. Constant segments. Okay. How do they vary? Alpha chain, they lack D, this one, D segments, D region. This particular protein is lack, there's no. And but they have the alpha chain, they have J segment, numerous J segments. So V and J constitute alpha, and the gamma chain, okay, and they gene cluster no D segments, the gamma, but rest they also have the gamma chain. So these are some of the differences, alpha and gamma chains, and how they looks like, the different functions on that one. Now they complex with the CD3, that the integral, what is CD3? We will see complementarity determining sites, that's the recognition of the antigen, by complementarity determination. As you mentioned before, the region, this is our type of protein, which is, and another one like this. So it forms a complement to this. And this determination by a protein, complementarity determining protein, and there are different types of, we call it in other words, we can put CDs, these are the protein, complementarity determining protein, we call it CD. There are different types of CDs are present in the blood, human blood. CD4, CD3, CD14, and CD72, like that. There are different varieties of the complementarity determining site. And they are not, I mean, they are not as a receptor, but they act with receptors. Just like the alpha and beta, the beta cells, I mean the B cells, which we studied earlier. And the beta B cell receptors cannot be activated by itself, but the signals is being transduced by item, which is present in the alpha and beta chain, the tyrosine kinase. The same way, the CDs, which is going along with the receptors. In other words, I draw a cartoon like this. If this is the receptor, R for receptors, receptor protein, but these CDs are present near to it, CDs, the clustering, I mean the complementarity, it's present nearby, and they are responsible for the signaling. These are the signal inducers. What will happen if the CDs are lacking? If the this particular protein is not there in the cell, the signal cannot be transduced. So this is very important, and along with it cannot act on its own, but it can act along with the receptors. The receptors and CDs, they present together. So in other words, we call it as the T cells and B cells, T and B cells, okay? They are the special army troops, right? Special army. They can what? They can identify the antigen, and also send out signal send out signals, okay, and also the relay the signal, send out the signal and relay the signal, and thereby it activate phagocytes and macrophages, okay. And the complex of this TCR2 and CD3, and they have the gamma and then epsilon 2 and epsilon 2. I mean, they form, all of them form a complex. This is the complex which I talked about earlier. This is being activated. In other words, this is called, in other words, ITAM, signal transduction, triggering molecules. Okay. And this chain, this epsilon chain of the TCR can bind with this one of these molecules, right? One of these subunits of this binds with the NK cell, natural killer cells. That's what they send out of the signals and then phagocytes and the NK cells, natural killer cells. And then the natural killer cells will go and bind, and then that will destroy that particular cells. So it will, the activation of these NK cells, that depends upon the T cells. When the T cell is activated, when it binds with an antigen, it can bind with the antigen. So this is the method of killing by T cell is different when compared to the B cells. B cells will bind with the antigen and they activate itself to form a mature, as well as it will synthesize more of antibody through plasma cells, and then it sends out the signal like antibody. But in the T cells itself, it will activate the NK cell, the natural killer cells, and thereby the natural killer cells will go and bind to the antigen which is present over there in the circulation and kill that cell. Okay, so we will see that mechanism, how it will do that one, right? So when you do the antigen recognition, how many antigens are there? There are plenty. So you need generation of diversity. There are different types of antigens, so you need generation of different varieties or diversities of receptor molecule of these T cells as well as the B cell. And it should recognize virtually any pathogen. As I mentioned before, like antibody, these receptor molecules also have a different varieties. So we have only 25,000 genes. But you have the pathogen or the recognition of different type of antigen, which is going to have a, you may have a millions of different types. We don't have that millions of genes in our system. As I mentioned before, one gene that will give two peptide, right? Remember, there's a differential splicing mechanism. That is the mechanism which is operating here. So that will give the same amount of genes, limited amount of genes, that will give different proteins. That's the proteins which is acting as a receptors, but the splicing mechanism. We call it the B, D, J, and C genes. B, D, J, C genes. When you see these combinations of these are the different combinations will produce more proteins. Is something like a number 1 to 10, right? So within this region of this number, you can, the whole lot of billions and billions of numbers and numerical algorithms, everything you can be built in, right? More number, 11, 12, 13, like, just if you go on to 100, how many numbers you can, innumerable, right? but the letters is only 10, just like a 26 letter of English alphabet. But how many words you are making it up? That's language, you are doing it, right? The same way, genes are the letters, are alphabets, and these proteins are the words, which is different combination of that. That's coming out. So don't expect, in those days, maybe 10, 15, 20 years back, they said one gene, one protein. That's a theory. But when they isolate the protein, they have got thousands and thousands of protein, but they don't find that many genes there. So in between, there is a gap. What is that? Because of the editing of this gene expression. Same gene. So it's nothing but the genes are like an alphabet, then the translation part and the transcription part, and there's the editing part, and everything plays a major role to produce a number of proteins, and that require different functions. So that concept is, fits in this situation as well. Okay. How they do it? There are different concepts here. One is the intra-chain amplification. Intra-chain amplification is one part where you have the random choose of VDJ segment. These are the gene segments, right? Combination that increases diversity as well as the geometrically it induces. Just like your Lego toys. I put in the other words. If with the Lego toys you have material, but you can build in a different bridge. You can make a plane or make a building or make an industry or make some other a vehicle like a car. Everything on the small bit and pieces of Lego toys. The same thing is here. You can building blocks with the genes, but the different combination of the VDJ segments, that's the limiter, and from that you can make sure the variable proteins. So limited genes, but the receptors are multiplicity, maximum receptors. So antigen specific receptors of the B cells and T cells, they are made through this intra-chain amplification. That's the one part. Okay. The light chain variable region, I put the light chain, heavy chain, like, light chain. Always the light chain is made up of V and J segments. V and J constitute for light chain. And the heavy chain of any protein there in this example, VDJ segment. V, D, and J segment. So here light chain is V and J because they're lacking a D. And the heavy chain molecules in this protein are the receptors, we have V, D, J. So one more protein, so it is a heavy chain. So that's the thing. So if you choose and a random joining, 25D chain is already there and 6J. So if you have a DJ, how much DJ is 25 times 6? You have 150 DJ molecule. So only 25D is existing, only 6J is existing. But when they combine with different types of D and different types of J, you can expect 150 DJ molecule in total. Do you understand? This is simple arithmetics. 25 different segments of D and 6 different segments of J. I want to know how many combinations it can make. 150, because each one will go on the D and J, right? It'll go. Do you follow that point? Good. And now there are 50 VH segments. So how much VH, D, and J together will form? So the calculation 150 times 50, that will give you 7,500 proteins or segments, the net. But when you think of how many genes make this protein, 25 plus 6 plus 50. 25 plus 6 plus 50. How many? 81. So the 81 genes that make different combinations to produce 7,500 proteins. Do you follow that? So if you want to have a receptors, a different type of specificity, 81 genes is enough. And that can combination with the different varieties, and that can produce 7,500 of what? This protein of VDJ. Individually it may vary, but the origin of the gene is same. Just like when I start my class, one gene, two protein, but here you have a different type of segments of genes of 25 different variety and 6 different variety, 150 in together, and then 150 with another 50, another type of segment. V segments, you have 7,500 different type of proteins. So this is the multiplicity, a different type of receptors. They use a different type of receptors here. He's calling here. Okay. Do you follow any question? This change, we call it as a geometric recombination. Geometric recombination. See, the one gene which is having two protein, that is differential splicing mechanism. Differential splicing, one gene. But here the different segments which will have combination, we call it as a geometric recombination, which will produce different varieties. Okay. The next one is another type is playing with junction. See, same type of protein here, another protein. Okay. So if they join together, it form one type of protein. If the junction of the two polypeptide is junction here, the disulfide, another type of protein, and here, and then it forms like here, it forms one side. See this. So these are the disulfide bridges. These are junctions of these two. Changing this junction, the disulfide bridge bonds, it gives more. Initially, we call it as 7,500, for example. So if you increase the 7,500, that playing with the junction, these junctions of this disulfide bridge, it will give you another maybe 10,700. Or, I mean, 100,000, 110,700, I put it. So different junction it can play, so you will change the shape of the protein. Do you follow that? How 81 genes, imagine just 81 genes, I told you how they form 7,500. Now the 7,500 proteins, but they combine, the combination of that may change at different varieties, more multiplication, different type of protein. Globular region, that's 100,000, for example. So it forms in more variation in a germ line, of germline repertoire. You know, that the storehouse, germline is the repertoire, is all different type of protein together. Okay. REPERTOIRE. Okay. It's coming up. And further diversity of this, you know, after this, of the junction, further there's been diversity of this coming from palindromic sequences. Palindromic sequences. That is, we call it as a P elements. Okay. So the palindrome, you know what is palindrome? Of the sequence in a DNA sequence, we have a T, C, G, T, T. What is the palindrome for this? A, T, C, G, T, T, right? So this is the one direction, right? When I write the same thing in the reverse direction, from this to here, it goes like that, right? It's a palindrome, we call it that. It's one in combination to the other, right? And that's the type of, when you find this type of sequence, it can also form a recombination process in this palindrome sequence. You have a insertion on this or deletion in the mutation, and they call it as again the mutation cycle, right? Mutation. And they also form a different variety and increase the variation. And they are very important, these variation of palindromic sequences that induces in the T cell receptor. So I'm just going to give the sequence from your textbook. If you want to follow, you just follow under the textbook. Just an example, I'm giving that of how the variations. This one as a CD3 molecule, items and everything, and this is the one, the TCR gene is spliced out, VDJ, how it forms. And this is a CD3 complex, this one on the figure 4-5, and the calculation of human V gene diversity, different type of in the table you will see. And these are the palindrome, how the palindrome is the D segment, how they join hairpin loops, and these are the receptor editing, which where you will find again the gene sequences being varied, and intra-chain amplification and somatic hypermutation, and then NH are the natural killer cell receptors. And this receptor molecule, where the antibody-coated targeted cell, where the activated receptors here, and this normal cell, this abnormal cell, and the locking MHC class 1, and these are natural killer cell, and this natural killer cell giving the signal to kill the antibody-coated cell or the infected cell. So this part, I will cover it up, the mechanism along with the MHC. So we will study the major histocompatibility complex on the next class on that one. So with this, just I want to give an introduction of this major histocompatibility complex. Yeah. Okay. Now I want to give you the homework for you. You can watch this video which just now in the class, okay, and then make a summary what we discussed in the class. Maybe you can pages 1 to 2 pages, 1 to 2 pages, that's enough. Make a summary and submit in WebCT or the Blackboard email attachment. Just the summary. Refer the book as well, but I want the summary of this, and when you come the next week, before that I can see your email attachment who has done this email. So if you want to discuss it, yes, discuss over that, as well as you can also call your friends, and then you can also inform to that. And before going there, I want to give the further animations a little bit that will give you more of the summary of what we discussed in the class. The membrane receptor antigen. This is the interaction of lymphocytes with the antigen, binding specialized to the cell surface. Antigen-specific receptors, functioning recognition unit. B and T cells are quite different receptors. That's what we observed, we discussed in the class. I'm sorry. Could you see that one here? No. Oh, in your PowerPoints. Okay. BCR, TCR, T cell surface receptor, B cell surface receptor, NK receptors, and major histocompatibility complex. So we have covered these 3 parts, and we will go through the MHC and then the next chapter as well for the next class. Antigen, B cell inserts in the Ig gene product into transmembrane segments into surface. We did study this, alpha, Ig beta, and also transduce the signals. Okay. These are the molecules, which is the alpha, beta, sulfide, alpha, beta. So this membrane IgM, it can bind with the antigen, but it cannot induce the signal. The signal is induced by these alpha and beta chain of this. They are called the item. So, and then the Ig receptors, Ag is the transmembrane dimer, each chain composed of Ig-like domain. And these are T cell surface receptors. Inner one constant, rather like a membrane bound Fab. That Fab is a fragment antibody binding, right? Both chains are required for the antigen recognition. these are some of the experiments. It's not much on this because the hybridoma, we will see about this because I didn't teach you about hybridoma technology. Yes, that we will go on to another couple of classes, then you will understand this. This is the, again, experiments. And these are type of TCRs, and we can, here the, actually this, it's not being translated in D, gamma, and delta chains, so it's going to small square, that's all. But other things that you remember the V, D, and J segments of each. And these are the, how the genes is being translated, how they are insertion which is going on. That one. And transcription and splicing from the DNA to, it is, I discussed in the class itself, right there. Again. So these are the CD3 complex on the T cell receptors, high chain and heavy chain and different segments which we did study. And these are the germline and recombinant and how they do in a germline sequences of the different V segment, J segment. And these are the palindromic and hairpin loops. I mean, how they amplify the signals. And this is receptor editing again. And then the IgM, IgD, different type of CDRs and, hypervariable region in a mathematical model. So they all put together the number of receptors which increase 7.5 into 10 to the power of 3 Ig, and then 200 light chains and 4.5 into 10 to the power of 3 TCR chains, and then TCR, another type of TCR1 into 10 to the power of 3. So you have different varieties now. Number of receptors is more, so recognizing different type of antigen. Further diversity, which we discussed earlier now, B cells and T cell, they undergo somatic hypermutation affecting the V region. Okay, and the NK cells bear a number of receptors, Ig domain, C type lectin, which we will eventually see on the next class as well, and this MHC molecules and everything. Okay, we will see this part next class about it. I want to go on to the concept.
The T-cell receptor on most circulating T cells consists of an alpha chain and a beta chain. Both the alpha and beta chain of the T-cell receptor are composed of constant regions in the membrane proximal region and variable regions on the membrane distal part of the molecule. As with antibody molecules, the T-cell receptor variable regions consist of 3 hypervariable loops at the tip of both the alpha chain and the beta chain. The third hypervariable region consists of a protein sequence that is encoded by V, D, and J gene segments in the case of the beta chain, and of V and J gene segments in the case of the alpha chain. Again, as with antibody molecules, it's the hypervariable loops in the T-cell receptor variable regions that are involved in antigen recognition. However, unlike antibody molecules which recognize native antigen, the T-cell receptor recognizes a combination of a peptide derived by intracellular proteolytic cleavage of the antigen, together with amino acids on the MHC molecule. The T-cell receptor is associated with a complex of molecules collectively called CD3. The CD3 complex consists of a CD3 delta, epsilon, and gamma chain, together with zeta and eta chains. The charges on the transmembrane parts of the T-cell receptor and the CD3 molecules permit association of these molecules within the plasma membrane. The zeta chains of the T-cell receptor complex are involved in signal transduction into the T-cell. An alternative form of the T-cell receptor consists of a gamma chain associated with a delta chain. This gamma-delta T-cell receptor, although not present on the majority of peripheral blood T lymphocytes, is found on large numbers of intraepithelial and gut mucosal T lymphocytes. The human MHC is called HLA, human leukocyte antigens. They're so called because they were first described on the cell surface of human leukocytes, although it's now known that for MHC class 1, virtually all nucleated cells express HLA antigens. The MHC gene locus can be divided into 3 classes, class 1, class 2, and class 3. In the human, These HLA antigens are found linked together on chromosome 6. The MHC class I region in man consists of HLA-B, HLA-C, HLA-A and HLA-G, whilst the MHC class II consists of HLA-DP, DQ and DR. Between the MHC class I and class II in man lie the so-called MHC class III genes. These consist of many different genes with diverse functions, many of which are not directly related to the immune system. However, some genes encoding products important for immune responses are found within the MHC class III region. These include genes for the complement components C4, C2 and factor B, and for TNF.
So I'll go through the MHC next class on this one. Do you have any questions so far in the class? Yes.